Hello everyone, my name is Vania Athalia and I'm from Universitas Airlangga. Today I'm going to present my research titled Potential of Phytase and Protease Enzyme on Tenderness and Cooking Loss in KUB Chicken Thigh Meat. Local chickens are well known for their rich flavor and complete protein content, but they still suffer from low productivity and too meat texture. Among all parts, thigh meat is more preferred by consumers because of its tenderness and better flavor. Improving the physical quality of this meat, especially its texture, could have increased the market value of local chickens, supporting the livelihoods of small farmers and meeting the growing demand for quality protein. KUB or Kampung Unggul Balitbangtan chickens are genetically selected local chickens developed to improve productivity. However, gaps in meat quality still remain. Phytase and protease are two enzymes that have been studied for their functional effects on animal nutrition. Phytase releases bone phosphorus, helping to increase muscle pH and water holding capacity. Protease breaks down structural proteins, which can improve meat tenderness. In this study, we aim to determine whether dietary supplementation of this enzyme can improve tenderness and reduce cooking loss, supporting SDG 2 Zero Hunger through feed innovation. This was a true experimental study using a completely randomized design with 28 male KUB chickens, each 7 days old, divided into 7 treatment groups with 4 replicates. Treatments included a control group, 3 levels of phytase at 200, 400, and 600 mg per kilogram feed, and 3 levels of protease at 300, 500, and 700 mg per kilogram, and the research was conducted at 3 locations. At the Poultry Experimental Cage 2 at the Faculty of Veterinary Medicine, Universitas Airlangga for treatment, and necropsy at the Animal Feed Lab at Universitas Airlangga and the Animal Product Technology Lab at Universitas Brawijaya for laboratory analysis. Here are the main tools and equipment used in this study. The Warner-Bressler shear force device was used to assess tenderness. The membered water bath was used to evaluate cooking loss. In addition, the following tools and materials supported the entire research process as cages, feeders, weighing scales, tender poultry vaccine, phytase and protease enzyme, storage containers, and general laboratory supplies. The research procedure followed these steps. First, cage preparation involved cleaning and disinfecting brooding dividers, installing lamps, and placing litter 1 week before the chicks arrived. For animal preparation, the DOCs were brooded at 33 Celsius with access to feed, water, and sugar solution. Chicks were weighed and graded at day 7 and day 14. Treatment started on day 15, where chicks received either control feed or enzyme-supplemented feed 3 times daily at 6, 12, and 17 GFMT plus 7. This lasted until day 90. On day 19, the chickens were harvested. Thigh meat sample size 5 times 5 centimeters and weighing at least 50 grams were collected and stored on dry ice. For tenderness test, shear force was measured using the Warner-Bressler device. For the cooking loss test, samples were cooked in a water bath and the percentage of weight loss was calculated. The data were analyzed using one-way ANOVA. If significant differences were found, they were further tested using Duncan multiple range test in SPSS. All research procedures follow institutional and national ethical guidelines and were approved by the Animal Care and Use Committee of Universitas Airlangga under the following certificate number. The tenderness test showed the following result. The T5 group, which received protease 500 mg per kilogram, had the lowest shear force, meaning the most tender meat. Moderate levels of phytase at 400 mg per kilogram and protease at 300 mg per kilogram also contribute to improved tenderness. However, the highest enzyme levels, 600 to 700 mg per kilogram, actually increased meat toughness. As for cooking loss, the T1 group with phytase 200 mg per kilogram had the lowest cooking loss, indicating the best water retention. This was followed by T4, which received protease 300 mg per kilogram. In contrast, higher enzyme doses were associated with increased cooking loss. Phytase works by improving phosphorus availability, which increases muscle pH and water holding capacity, softening the meat fibers. Protease contributes by breaking down myofibrillar proteins, reducing shear force. However, when enzyme levels are too high, they can damage structural proteins, reversing the beneficial effect. These findings are consistent with those reported by Samart and Oyeko. Low to moderate enzyme supplementation help maintain muscle structure and pH balance, enhancing water holding capacity. Phytase stabilize the muscle's mineral composition, and protease improve the binding of the water in muscle fibers. On the other hand, oversupplementation causes cell damage, increasing dip and cook-out loss. This aligns with studies by Maynard and Arsha. Based on our findings, tenderness was optimized at 500 mg per kilogram protease and 400 mg per kilogram phytase. Cooking loss was minimized at 200 mg per kilogram phytase and 300 mg per kilogram protease. Therefore, targeted enzyme dosing can improve meat quality and reduce post-harvest losses, offering benefits to both producer and consumer. For future research, we recommend evaluating combined enzyme use, exploring dose response pattern, investigating pH and water holding mechanism, and conducting validation trials in commercial farm settings. We would like to thank Universitas Airlangga for funding this study, along with the Animal Feed Lab and the Animal Product Technology Lab for providing the facilities and technical support. We also extend our appreciation to the research team for their valuable contribution. That concludes my presentation. Thank you for your kind attention.